Welcome, everyone. My name is Adriana Matos Nieves, and I have the distinct pleasure of welcoming you to the René Salazar Speaker Series. We are joined by Felix Moronta Barrios today and three of our cohort members from the BSSP program, Maggie, Jada, and Marissa, who will be facilitating this conversation. Before we kick off, I just want to give you a bit of a introduction as to what the René Salazar Speaker Series is meant to do. It's actually meant to highlight researchers from within different scientific paths and disciplines, just so we can learn more about our fellow brothers and sisters in science, foster collaborations amongst high-level researchers and younger researchers like we're going to see today, highlight role models from varied personal and professional background experiences, promote opportunities to network and opportunities to do research. So we hope that you have a very engaging discussion and conversation today. LatinX at Broad specifically has the mission of building community amongst Brodies and offering opportunities for connections, learnings, and belongings. We have a very vast calendar of events, especially for Hispanic Heritage Month coming out. So please feel free to reach out to me directly or anybody on the steering committee if you want to get involved with that. And without further ado, I have the distinct pleasure of introducing Felix Moronta Barrios, PhD, who is a senior program assistant in the Regulatory Science Group at the International Center for Genetic Engineering and Biotechnology, ICGEB, where he supports building and policy development in biosafety, biosecurity, and the governance of emerging biotechnologies, including gene drives and synthetic biology. With a strong focus of advancing science-informed regulation and responsible innovation, particularly in the Global South, Felix contributes to international initiatives that strengthen regulatory frameworks and promote equitable access to biotechnology. In addition to his role at ICGEB, Felix served as co-chair of the Global Young Academy for the 2023-2024 term, leading global efforts to amplify the voice of young scientists in science diplomacy, policy, and education. He has been active in the international advisory panels, including the World Health Organization's Working Group on Gene Drive Research, and has collaborated with UNESCO and the Inter-American Institute for Cooperation on Agriculture on issues related to biotechnology governance. Felix holds a degree from the Central University of Venezuela and earned his PhD in biology from the University of Alicante in Spain. He is passionate about science communication, public engagement, and the role of sustainable science development. Thank you so much for being here, Felix.
Now, thank you, Adriana, for that fantastic introduction. Thank you so much, and thank you so much for the entire team for the invitation. It's an honor to be here and to talk with your audience about, well, my work and the history of how I arrived where I am now. And this is just a little part of the professional life of a scientist. So if you permit me, I will propose to start my conversation just sharing some slides about my institute. I'm interested for you all to know what my institute is about and some other insights about the work that we do in my group. So at the ICGEB, this is the International Center for Genetic Engineering and Biotechnology, and we are an intergovernmental organization that is working in the intersection between research, training, and technology transfer in life sciences, with the goal of promoting sustainable development, the sustainable global development. And we began as a special project of UNIDO. This is the United Nations Office for the Industrial Development. And then almost, 40 years ago, 30 years ago, we became an independent organization, fully independent. But still we operate within the United Nations systems. We follow many of its administrative procedures. And as an intergovernmental organization, we are made by the network of almost 70 member states, countries. And our laboratories are hosted in Africa, in Italy, where I am now, in South Africa, and also in India. And yeah, I will be happy to see many other Latin American countries and from Africa and from the Global South to join this group of countries and to complete this map. And the ICGEB is governed by a board of governors, no? And with one representative of each member state. And this board meets every year to help guide our direction of the institute. And in many ways, this functions like a mini United Nations, like a General Assembly of the United Nations, but for biotechnology, bringing diverse national perspective together with a spirit of collaboration. And our work is organized around 6 core pillars that you can see here in this list. And in addition, we focus on cross-cutting priorities such as gender and just South-South cooperation, and with a strong focus on the least developed countries. And our instrument of actions are these 6 that you can see here to deliver our mission. So the first one is about research in our laboratories, and this is just a very short summary of our diversity on the research part on infectious diseases, non-communicable diseases, and these are the groups that are distributed between our 3 components in, again, Italy, South Africa, and India. Medical biotech, industrial biotech, and also plant biology and biotechnology. And in this last group is where my groups has been assigned. Okay. So, and we also work with a council of scientific advisors that they provide with advice, so how to run our institution, and it's made by well-renowned scientific personalities, including Nobel Prizes. And then the other 4 instruments of actions are fellowships. This slide is not updated. I'm sorry for that, but constantly we have many calls for fellowships, PhD and postdoc fellowships, and also for short-term fellowship for people from our member states to come, national from our member states to come to our laboratories to perform a part of their research. We also help to organize, providing financial help for researchers, again, in our member state to organize meetings and courses. And also we provide grant research for researchers located in our member countries to perform their research there with a relevant context, in national context. We also have a tech transfer and biotech development unit to develop biosimilars with pharma compliance laboratories and premises. And the last but not the least instrument of action is technical assistance. And again, this is where my group is working, the technical assistant for the regulation of biotech and its product. So this is the ICGEB Regulatory Science Group. We support countries and organizations, agencies in the safe regulation of biotechnology. And we work to build and also strengthen systems that can effectively identify, assess, manage, and monitor biotech products, especially in the face of the emerging biotechnologies. And we organize our activities into 3 main areas: capacity development, knowledge and policy engagement, and diagnostic and applied projects. In the case of capacity development, most of our work is focused on institutional capacity development. For instance, developing or updating biosafety legislation and regulatory systems. Some example, and also strengthening institutional policy for the safe use of emerging biotechnologies. This is some example of our recent projects. and also the individual capacity building activities. We provide, organize and provide international workshops on environmental risk assessment of genetically modified organisms. And we have developed, and we are developing constantly, an ICG e-learning platform. In the area of knowledge and policy engagement, we make desk research. We produce literature reviews on biosafety legislation. And also we produce great literature. We write reports for UN agencies, as this one on synthetic biology, or policy briefs to address to international and global leaders. And also we participated in several policy dialogues, for biosecurity governance and strategic foresight, or more recent, this more esoteric part of biotech that is called Mirror Lives. And a small part of our activity is devoted to diagnostics and applied projects. This is something that we have recently implemented in Africa for strengthening the surveillance and epidemiology of public health laboratories in Africa. And this is my group. We are today just 4 groups. This is my supervisor, Wendy Craig, and my other 2 colleagues. And we rely on our extensive network of collaborators for filling any gaps that we might have in our projects. So that's it. That was short and quick, but I hope that provides you guys with a very broad idea of what my organization is and what more or less we are doing in my group. And I will be happy to discuss for the rest of the time about what I told you or any other issue about biotech and recent interesting topic or future interesting topic, of course, of biotech.
We are joined by 3 students from the Broad Summer Scholars Program, and they have prepared a list of questions ahead of time to facilitate the conversation. So Maggie, would you kick us off, please?
Yeah, so the first question we'd like to ask is, what first sparked your interest in biology, and how did that interest grow into a career in science and policy?
Yeah. Okay. I mean, many things. I mean, this is something now in retrospective. I can think that, I mean, I always was fascinated by living things and by science. And perhaps it was not unique for me, because I think that any child is fascinated by living things. But I remember, to be very mesmerized of how plants grow and how insects behave and how our bodies work. And I remember I was watching Animal Planet and Discovery Channel instead of MTV or Cartoon Network or something like that. But that was something that I bring with me for the rest of my life. There was an interest that was growing so much, and this innate interest by science. And well, then I decided to study biology without really knowing about what kind of work a biologist does, honestly. But then during the career, during the university, I discovered that I was in the right place. So there was a combination of things.
Thank you for your answer. Our next question is, can you tell us about like a moment in your life when you ended up realizing that you wanted to make science useful outside of the lab?
Yeah. Okay, yeah, thank you, Marisa. And again, it was not, usually it's not an eureka moment, the one moment that you say, oh, this was, no, it is a combination, but the moment arrives, when you say, okay, something need to change here. And when when I finished already my career, I was a graduated student. I started to make research, in a laboratory, thinking with a scientific method and making a hypothesis and experimentations and blah blah, and having hypotheses and proving the theories, all the history about science, and then publishing my first paper. And nobody reads my paper. Absolutely not. So that was extremely technical. And it was, of course, addressed to my peers. So I was trying to tell my family, to my wife and my mom and my dad, what my work was about. And I didn't know how to do that. And they were bored because of my histories of experiments, of failed experiments, or suddenly my paper. So that my work, years of work and nights of work, and well, many things were not reaching the people, the society. That was a norm. It is a norm. Absolutely. It is a norm in our laboratories. And then I realized that I was not happy with that situation. Absolutely not. And I couldn't wait for anyone to solve that situation. So I started to take training in science communication. But in that moment, when I realized that my research was not reaching the public, I said, okay, I realized that I would like to make something for that to happen.
Thank you. That definitely shows a lot of your perseverance. So our next question is, what skills from your research career helped you transition into international work in science diplomacy?
Yeah, again, a combination of skills, absolutely. And that transition was not from one moment to another. It was not from one day to the next day. It's a transition that is built step by step, slowly. And well, I already told you guys that I started during my PhD to take training on science communication, and that was extremely helpful, extremely helpful. That was the first thing. I mean, that was the basis, the foundation for my transition. Science communication, get trained in science communication. I mean, taking courses and certifications in science communication. That was life-changing, totally. And then I started to write a blog. So trying to put in practice what I was learning in science communication, and I opened a blog. I started to work on my personal branding. And I cannot see at this moment in my time, because that part was 10 or 12 years ago. And now I don't want to read what I wrote in that time. I don't want to see my former website because I don't like it anymore. Of course, we all change and we evolve, but that was extremely helpful, extremely helpful. So that put my name there in the stage, that started to position myself as it was not a leader because I was extremely young, but as an emerging leader, let's say, no, and making it to start expanding my network, So it was that science communication, then personal branding, absolutely. And these pieces of decision or action then open the door for other things, like I was accepted for to take a training on scientific advice, and then a training on science diplomacy, and well, the rest is history. But the basis was science communication and personal branding.
Thank you so much. There's a question from the audience. So
Yeah.
they ask, where do you think is the most interesting environment for a young scientist to grow right into?
Hmm. Yeah. Wow. Okay. For young scientists, well, young scientists, absolutely, a research laboratory is mandatory. Absolutely, no doubt. It is like that. This is our way of life with all the imperfections of academy, etc. But it's the way it is, and it is okay. But it needs to be complemented. Absolutely. So for building a socially relevant career, because I think that this is key and this is important to build a social relevant career. It is important to devote and dedicate time to civil organization, something that connects you with the real problems that are not the problem from your family or your neighborhood, are more complicated and complex societal problems. So being volunteering to civil society organization, providing your skills as a geoscientist is extremely helpful. So, and that will enrich your training, your development as a early career researcher. I'm including here in civil society organization, like organized students or science academies, that they are also with a very good connection with society. And of course, things that are not related to science, but still science has a component there. Environment, something like that. Food, the use of the land. There are many things. Yeah.
All right, wonderful. Thank you. So next, what does your work look like on a day-to-day basis when helping countries implement biosafety measures?
Yeah, my day-to-day basis. I don't want to say that it's boring because it is not. But honestly, the most of my days, I am in my desk, in front of my computer, writing emails, reading reports, and writing reports. But I enjoy it so much. It's not a mechanical work, absolutely not. I need to put there huge amount of intellectual effort because I need to be very well educated on emerging biotechnologies in the more complicated as molecular aspect of the last genome editing technologies because I need to communicate that to policymakers and decision makers, and I need to do it in a language that they understand. But because of um, my training and my formation as a scientist, I can understand both worlds, the academic, and I have learned to understand the policy work. So I am there, as the bridge between these two worlds. This is 70, 70, maybe 80% of my time. The rest is extremely exciting because it's traveling. I work in an intergovernmental organization and our training because I am a trainer now too, and scientific advisor. I need to go to the countries, to ministries, governmental agencies, to other international fora, to other UN agencies to provide our work. So I am in my desk, reading, writing, mm, implementing projects from my desk, and traveling.
Thank you for answering that question. Our next question is, in your opinion, how can genetically modified organisms or GMOs both help and even potentially harm biodiversity?
Okay. Yeah. I mean, it is not my opinion. It's scientific evidence here. It's scientific evidence, and GMOs can, for instance, help reducing the need for pesticides. And this is something that has been very well established and measured, that the use of pesticide has been dramatically reduced thanks to the GMOs. Just think that many of them are resistant to pests, so it is not needed to apply the same amount of pesticides. Or, for instance, now they are developing crops that are more resilient to climate stress, and this is something that, of course, is going to provide benefits to the biodiversity and environment in general. These are just two examples. We need to think that the GMOs are the genetically modified organism. In the case of plants, they are improved plants. This is made by breeding. It is plant improvement. So we are taking advantage of genetic knowledge to improve our crops. But of course, as any technology, any human technology, there are associated risks. There are risks in building a bridge. There are risks to take a plane and travel. There are risks to when you turn on your kitchen. There are risks there. The thing is how to manage those risks, no? How to cope with those potential risks. And biotechnology do exactly the same. No? It is important to maximize the benefits of GMO and at the same time minimizing the potential harm. For instance, if a GMO is not well managed, let's say, well, it could affect absolutely wild species, wild plants, or the soil microbes, or perhaps more broad ecosystem. It is not well managed. But this is something that is not happening. GMOs is one of the technologies of the product that is more regulated and regulated in the world. So far, there has been no any important harm to biodiversity, at least in the scientific literature. There are testimonies and any other sources of information, but in scientific literature, there are no.
Thank you. We also have a question from the audience. So could you describe an example of a case in which you and your team managed to make a difference in a country's policy? And in addition to that, what was the process like? What were the challenges like? And what was the time span?
Yeah. Yeah, I can highlight one particular example. It was during the COVID pandemic. I was working in a project for modernizing the biosafety regulatory framework of the Republic of Panama in Central America. The thing is that it exists an international agreement, and the international agreements are legally binding documents. So if a country signs that international agreement, that country must put in place laws and regulation for that agreement to make it happen in that country. So in the beginning of the year, when it was 1999, the 2002, the Cartagena Protocol on Biosafety was signed and established by most of the countries of the world. This is an international agreement that establishes rules for the transboundary movement of GMOs. Okay, that's fine. Now the countries during those years, most of the countries, they draft their biosafety laws and they started to implement the Cartagena Protocol. We are talking about 2002, year 2002. So this is 23 years ago. In that time, genome editing didn't exist. It was not discovered. CRISPR and many technologies that are now in almost any molecular biology lab. In that moment, no clues about that technology, no clues about synthetic biology, and many other things. So now the countries are working, they are regulating their biotech product with an agreement that was signed predating those scientific discoveries. So they need to update their national laws. So that was the call from many countries. Panama was able to mobilize resources and to bring us in place for helping them to modernize their regulatory framework. It was a fantastic experience working with national and with officers from Panama. The challenge, for me was unexpected, and is how difficult it for different national agencies to sit together and draft a document, that it would be good for their country. What does this mean? I mean, biosafety committee, from a country, or the biosafety competent authority usually is made from different people from many different ministers. A group from the Minister of Agriculture, a group from the Minister of Environment, Ministry of Trade, Commerce, Ministry of Health, so on. They need to sit together to establish this regulation, and that's difficult. They all come with their mindset on agriculture or health, and that's the idea, of course. But they need to, the knowledge needs to permeate between these silos, national silos, and that was something that was a learning experience for me. But this is not that easy.
Thank you so much. What's one example of a successful biosafety project you worked on that made a difference?
Yeah, I mean, because I work building capacities or strengthening capacities in biosafety. For instance, we recently implemented a project of regional workshops on biosafety, particularly in the environmental risk assessment of genetically modified organisms. There were 5 regional workshops in the world covering all the continents. And each of the workshops in all the continents were tailored to the problems of that region. So we provide with the theoretical part, of course, of the risk analysis, with risk assessment, and so on. And then we want the attendees, and they are a mixture of regulator, scientists, farmers, etc. We want them to analyze hypothetical cases for assessing potential risks of a genetically modified organism. For instance, we went to Central Asia, where wheat is produced, is the main crop. So we developed an hypothetical genetically modified wheat adapted to the environmental conditions of Central Africa. In the workshop for Oceania and the Pacific Island, we developed a case study of genetically modified rodents, mice, as an invasive species in island. And then for Africa, we developed a genetically modified banana, in hypothetical case. Recently in Brazil, we developed a case study on genetically modified mosquitoes for fighting dengue and other context-relevant diseases. So, and this very large, multi-year and international global project was extremely successful. I mean, because we were able to train a diversity of people, and our hope is that they implement what they have learned during our training in their countries.
Maggie, apologies. I think you're muted.
All right, thank you. So our next question is, how can science help bring countries together or solve global problems like food security or climate change?
Yeah, thank you, Maggie. You know, there has been precedents on how science can improve international relations. Typically, during the Cold War, for instance, there were big scientific projects that helped release a little bit the tensions for the establishment, I don't know, of research institute for s- space exploration, for instance, was another topic. And there are several examples of how they can help alleviate problems. Because science provides a common language, not something that can be understood, and also a shared evidence-based approaches. Of course, the political tension are not solved, absolutely not, but science helps to create spaces for dialogue and cooperation, and that's amazing. For instance, here where I live in a little town that is called Trieste in Italy, and we are in the border with Slovenia in the northeast of Italy. And this little town, little city, Trieste is the city in Europe with the bigger number of researchers per habitant in the entire Europe, and that's amazing. And that's because here there are several international scientific organizations like mine about biotech and genetic engineering, and there is another one on physics, and there is another couple of international organizations and UNESCO offices and so on. And that was because this was the last city before the communist bloc. Uh, Now, when Slovenia was part of Yugoslavia and the communist country. So, and here in Trieste was a point of to encounter, no, the first and the second world, no, the capitalist and communist blocs from the world were here in Trieste. So, when this city was developed, uh, so much thanks to that scientific bridge there. So these are just examples. Now, for the current global problems, like food security, that is sadly a never-ending problem, and now more concerning the climate change problem, is more or less the same thing. We need to think that we are in a common space, that is the planet Earth, and no a single solution, no a single country would be able to solve any of these problems. And there is need for a more diplomacy and science diplomacy approach for solving this problem.
Thank you for that answer. Our next question is, why, in your opinion, why is it important to share science in people's native language?
Yes, and this touched me inside. No? That's because you are noticing that English is not my mother tongue, and I have been one of the affected, no? Because this strong limitation of the language. I must recognize, however, that it is extremely beneficial to have a common language for sciences. Absolutely. So I very much support the use of one single language uh, for having these global discussions and trying to solve the global problems, no doubt. And yeah, in the past was Latin, Greek, French, whatever language. Now it's English, and it is okay. We need to consider, however, that population, most of the population doesn't speak English at all. So if we want for the scientific evidence and the global discussion to arrive to society, we need to talk in their languages. Absolutely. Everyone should be included in discussion, especially when they are the affected community with the problems or the potential deployment of technological solutions. Yeah, uh, they absolutely need to be reached by context relevant scientific information, including no, in their native languages.
Thank you so much. What advice would you give to students who are interested in both science and making a global impact?
Stay curious and stay open. Stay open and yeah, these students that are interested in both making a global impact and in science, yeah, stay curious, stay open. The foundation of science and the values of science must be protagonist during your entire lives, guys. Absolutely. This is something that the scientific method should be the guiding light in all your actions. It is important to adapt this career. Science is extremely changing. You need to adapt to any challenging or nice situation. So adaptation is key. And don't be afraid to step outside the lab. There are several options, or there is life beyond a lab bench. And this is built again with, well, strengthening your soft skills, beyond science, yeah, expanding your network and being active and doing things that are socially relevant.
All right, thank you. So not too long ago, you and your colleagues published a paper titled Navigating Biosafety Regulatory
Mhm.
Frameworks for Genetic Engineering in Africa, a focus on genome editing and gene drive technologies. Could you tell us a bit about what you and your colleagues found?
Yeah, sure. Thank you. Thank you for highlighting that paper. It wasn't that by the late 2024, we published this paper when we analyzed, the regulatory frameworks across Africa. And we looked at how different African countries are approaching the regulation of this emerging biotech. And we found that there is a lot of diversity. There are countries that are doing so well, no? Countries that are well advanced in the regulations there, but others are still with no regulations. So there is a huge difference there and a very heterogeneous regulatory environment there. But the good thing is that they all recognize these gaps and this challenging, and they are all open to this to perform or to strengthen regional collaboration in making sure that all the regulations are both science-based or science-informed, and at the same time that are again locally relevant for the problems and to be relevant for the context.
Thank you for telling us about your paper. Uh, our next question is, why might some people be unsure or maybe even afraid of gene editing? And how can scientists and the governments help the public feel much more informed and safe?
Hmm. I mean, yeah, thank you. Thank you for your question. It is completely normal to be cautious and to be afraid of new technologies. This is something that is in our nature. When the electricity was invented, there were group of people opposing electricity. There were anti-electricity groups. When the train was invented, there were anti-train group of people and protesting in the street because they didn't want trains in their countries. And then, yeah, and so on. With the horses, there were anti-horses group of people. With the car, there were anti-car group of people. With any technology that is disrupting, that is completely different, that is no precedence in the history, there will be opposition. And biotech is not the exception. It is another human invention and technology. So with genome editing, with gene editing has been exactly the same. First with GMO, 30 years ago, and still there is some, yeah, so no, there is still an important opposition to GMOs. And now with gene editing, it seems that things are going to be better for the technology, for the biotech, but still some resistance out there. And I think that the key things here is transparency, explaining how these tools work, what kind of safeguards are in place for avoiding harm, and what kind of risk and benefits are. And building this trust takes time, but it starts with listening, not just explaining this dialogue. And what I am seeing now is that hopefully we have learned from the mistake from the past, the mistake that were done during the GMO era 30, 20 years ago. For instance, that the products were more designed for the benefit of farmers, so the consumer were not feeling that this kind of technology are a benefit for them. And this with genome editing is changing. Now, for instance, there are developing crops that are biofortified, so we can get more nutrients in our foods, or the climate resilient crops. There's some benefit that we can see, the benefit for everyone, not just for the farmers. So yeah, exactly. So more an expanded benefits and also transparency.
Thank you so much. How do you explain complex science in ways that help policymakers and everyday people understand what's at stake?
Yeah. This is a tricky one, and it's not that easy. And this is a learning process, and it is an evolution, how to explain these complex issues to policymaker and technical, complex and technical. I'm still learning because, again, policymakers are not a homogeneous group. They are extremely diverse, very heterogeneous, intergenerational, etc. So it's extremely challenging. I try to use analogies, histories, visuals whenever possible. What I don't use is jargon, scientific jargon. I try to avoid it as much as possible. And I try to focus on in the so what. So we have a new technology. So what? No? So what does this mean for food on the table? What does this mean for disease control, for pest control? So that again, that connection with the social part, the social relevant of that technology. But again, this is something that need to be adapted. There is no one size fits all recipe for explaining these complex technological issues for policymakers.
Thank you. How do you believe your work has grown you as both a human and a scientist?
Again, let me see. If my work, could you repeat?
Definitely. How do you believe your work has grown you as both a human and a scientist?
Okay, yeah. Well, being perfectly honest, doing this kind of work is, I mean, separating totally your professional and personal life, it's not possible. It is a mindset. Being scientist is a mindset. Absolutely, I'm sure of that. Uh, this is something that is with you for your entire life, and it dictates so much your way of life, your decisions, your way of, I don't know, to raise your child, the way you engage with your society, your neighborhood, your friends, etc. It's something that it becomes part of your person. So from a personal life, uh, I would say that my work has improved my personal life. I have learned many skills that I try to implement in my personal life on diplomacy, on management, trying to cope with different group of people, intercultural discussions. This is something that it is beyond my work. So what I have worked there, then with my intercultural group of friends, I try to take some bites and from what I learned there to my personal life. In my professional lives, again, it is a learning experience. This is learning by doing, just managing projects and trying to implement international projects uh, is something that always enrich your toolkit, no, for being a good worker, let's say. And something crucial always is your network. Is your network. So attending conferences is extremely important, but also your online presence is not trivial. That's not banal. Absolutely not. Having a good online presence and being professional in your online life is something that can improve and change your professional career.
Thank you for your answer. I have another question. So, how do you make sure that like new science, such as GMOs, end up respecting what people in a specific country believe in or respecting what they care about?
Respecting. Okay. Okay. In this case, I mean, I cannot answer in the place of the developers. I'm not a developer of GMO. Absolutely not. So again, my work is try to help governments, to make a safe use of or how they can ensure that the GM crops are safely used or something like that. So it is up to each of the government to decide if a biotech application is aligned or is not contradictory or it's not going to create tensions with their population. So there are socioeconomic aspects that from any technology, again, is not specific for biotech. There are socioeconomic aspect, and I'm including here, for instance, ethics, well, and many other things that the governments need to consider. Our role as a scientist are science-based advice or evidence. And yes, we need to give responsibly advice on how to apply science. But the part of socioeconomics is not, according international agreements, are not part of the scientific risk assessment. So this is part of the decision makers need to consider other sources of information. For instance, the ethics, religious people, indigenous people, minorities, etc. So that's the role of a decision maker, to keep an eye on all this spectrum of sources of information to then take a decision. Science is just one little part of the source of information. And all they are important. It's not that science is more important. No, no, no. They all are important.
All right, thank you very much. So lastly, why do you think it's important to engage with communities where biotech work is being performed?
Well, but basically because they are the ones most affected. No, they are they would be the affected communities. For instance, I am recently very much not involved, but I'm working. I'm developing literature reviews on gene drive. Gene drive is a technology that is looking for modifying, and listen, this is crazy, modifying wild population of mosquitoes. This is something that has never been done. Absolutely not. To modify a wild population, genetically modify a wild population of mosquitoes, this is extreme. Absolutely. And they are looking for modifying this mosquito for ending or help ending malaria. Malaria is one of the more deadliest diseases in the world. So there is a public health issue that genetic intervention is looking to solve. No? This is gene drive. So the potential release, because this is not happening yet, the potential release of these genetically modified mosquitoes with gene drive are going to happen in rural parts of Africa, for instance. That is the most affected part of the world with malaria. And what they are doing from 10 years ago, they are already working with these communities, with this local community, to co-develop the inception of the experiments even. So this is part of the they are trying not to repeat the mistake from the past. No? For instance, with GMO, they suddenly arrive. We have this GMO and it could save your life. Blah blah. Eat it or plant it. No, this was an extreme fail mistake, an epic fail. Absolutely. Now what they are doing, they are sit in the same table from the inception of the experiment with the local communities. Let's try to co-develop the experimentation. What are the needs? What are your conditions for a potential release of mosquitoes in 20 years? And they are engaging and working with them now, with the communities. And I think that that's the way it is for that kind of extreme interventions.
Well, Felix, thank you so much for this wonderful conversation. And I want to thank our amazing panelists, Jada, Marissa, and Maggie, for facilitating the conversation as well. I learned a lot, and I know that they did as well. Everybody, thank you so much for being here. And if you get a chance, please let us know what you thought of this talk by answering the survey that I just dropped in the chat. Thank you so much, everybody. Have a great day.
Great. Thank you, Adriana. And thank you, Jana, Maggie, and Marissa. Thank you so much. I have enjoyed this conversation.